Welcome, everyone, to Let's Talk Science. Our speaker today is Bill Agosta, a fellow member of the Let's Talk Science Committee, a biochemist who received his doctorate from UC Berkeley and subsequently taught at Rockefeller University, from which he retired in 1998 as Professor Emeritus. Over the course of his career, he established a reputation for research in organic chemistry and for contributions to science education. I perused the list of 273 publications credited to him, obviously intended for his peers in biochemistry, not clinical psychologists. I could barely pronounce the titles of those things, let alone understand them, but as you will find, he is a very lucid and very clear explanation of the topic for today, which is chemical communication in the biological world. Please welcome Bill Agosta.
Well, some of that was right. Today we're going to take a journey into the invisible world of chemical communication in biology, a world that exists all around us, often unnoticed, yet profoundly influential. While we humans tend to think of communication in terms of sight, sound, and touch, many other organisms rely heavily on taste and smell, typically mediated by airborne or waterborne chemicals. The ability to detect and respond to chemicals in the environment appeared early in evolution and is now found in everything from bacteria to elephants. Among the most intriguing of these signals are pheromones, special chemicals secreted by organisms to communicate with others of their own species. Whether it's ants following a scent trail to food, moths finding mates over miles of separation, or a newborn human baby responding to the scent of its mother's breast, pheromones play a powerful and often surprising role in shaping behavior. The idea that animals respond to chemical cues isn't new. The ancient Greeks knew that secretions from female dogs in heat were attractive to males. A beekeeper's manual from 17th century England noted that the odor of an injured bee would attract other bees, anger them, and cause them to sting. But the story doesn't stop within species. Nature is also full of interspecific signals, chemicals exchanged between different species to influence their behavior. Plants call for help from wasps when attacked by caterpillars. Parasitic fungi lure their insect hosts into deadly traps. Even humans respond, consciously or not, to the smells of other creatures. So we'll explore how these chemical signals work and how they shape the lives of organisms, including ourselves. To locate us scientifically, this is a topic in the general area of ecology, the study of how living organisms interact with the world around them, that is, with their environment. Among many other things, these interactions include chemical signals that we are going to discuss. So our subject is generally known as chemical ecology. We want to make an important distinction here. There are a few dozen chemicals necessary for life as we know it, but these are not our focus today. Our subject is rather the special chemicals with very limited distribution that serve precise functions in specific organisms. For example, nicotine is a plant-protecting compound found in reasonable amounts only in the tobacco plant and a few close relatives. There's no nicotine to be found in roses, tigers, monkeys, or daffodils. It's toxic and bitter tasting, and it repels herbivores, which are organisms that feed on plants. And so nicotine protects the tobacco plant from being eaten. Other plants have their own protective compounds that function similarly, but are not chemically related to nicotine. We'll come back to discuss interspecific signals later, but first we want to talk about pheromones. About 100 years ago, behaviorists became interested in the general question of how male and female members of a species find each other for mating. Insects were particularly easy to work with, and early studies showed that a receptive female silkworm moth releases a signal to attract males. It turned out that this attractant also caused the female to flap her wings. In nature, this presumably allows the male to locate the female more readily. Chemical fractionation of the female secretion was followed biologically by checking purified fractions for whether they caused more or less with female wing flapping. After 20 years of work and the sacrifice of over 500,000 female moths, the structure of this chemical attractant was finally determined in 1959. Chemical ecology was underway. In the 1960s, an entomologist, that is, a scientist who studies insects, an entomologist, E.O. Wilson, showed that ant scouts release chemicals from the tip of their abdomens to lay down trails for their nestmates to follow to sources of food that the scouts have found. Different species of ants use different trail chemicals to keep their trails and food sources secret from one another. Other signals within an ant species carry a variety of messages. For example, when two ants meet, they touch their antennae onto each other's body. In doing this, they detect chemicals that reveal not only each other's species, but also whether they belong to the same nest and whether they are workers or soldiers or have some other job in the colony. For the last 40 years of the 20th century, work with insects revealed the existence and chemical structures of many sex-attractant pheromones, as well as signals for many other purposes. The final signal a bee or ant sends to its nestmates comes after death. As the dead insects begin to decompose, it emits signals that tell nestmates to remove the corpse from the colony. Other signals concern interactions among living organisms. The queen in an ant colony releases a chemical signal that prevents the worker ants surrounding her and tending to her needs from developing into egg-laying queens. Should the queen die or disappear, the pheromone, of course, disappears, and some of these workers will then begin developing into queens so the ant colony can carry on. Soldier ants, perceiving an intruder at the entrance to the colony, release an alarm pheromone that causes increased alertness and aggressiveness in the colony's worker ants. Bark beetles provide another sort of signal. There are about 6,000 species of bark beetles worldwide. Many of these species attack trees in large numbers. There they mate and lay their eggs in the bark. When the eggs hatch, the larvae feed on the living tissue beneath the bark, and so weaken or destroy the tree. On finding a new suitable tree to exploit, bark beetles release an aggregation pheromone to attract other members of their species from afar to join them to mate and lay eggs in the attractive new setting. When a tree is full of bark beetles, the resident beetles release an anti-aggregation pheromone telling other beetles to stay away. In this way, they can exploit the tree without killing it from overfeeding. In general, both aggregation and anti-aggregation pheromones are mixtures of small, volatile chemicals synthesized by the feeding bark beetles. Bark beetle pheromones provide an excellent example of how we humans can make use of pheromones to control insects. The U.S. Forest Service employs bark beetle attractants to bring beetles to a chosen site for their destruction, while it employs the anti-aggregation signals to keep beetles away from trees that are to be protected. Most early investigations were done on insects, which were particularly easy to work with, but eventually behavioral and then chemical pheromone research spread to other classes of organisms. We looked first at a pheromone common to many different bacteria. Even these simple one-celled organisms use chemical signals to communicate with other bacteria of the same species. The particular message we will discuss asks the question, how many of us are there? And this activity goes by the name of quorum sensing. You will recall that in assemblies or societies, a quorum is the minimum number of members that must be present at a meeting for the proceedings to be valid. You will see in a moment why the bacteria are questioning the presence of a quorum. Like people, some bacteria behave differently depending on whether they're alone or in a group. Some people are well enough behaved when alone but can become violent in a large group. The same is true of bacteria, and a good example is a common bacterium known as Staphylococcus aureus, commonly called Staph aureus or even just Staph. Staph aureus's question about how many of them are present turns out to be very important for human disease. This is a bacterium commonly found on the skin and in the nose of about 30% of humans. Usually it's harmless, but depending on the answer to the question it asks, it can become a source of serious infection. In this country, it is a leading cause of hospital infections that can lead to pneumonia, bacteria in the blood, and sepsis. On top of that, some strains of Staph aureus are resistant to virtually all available antibiotics. Like many other pathogenic bacteria, Staph aureus uses quorum sensing to regulate its production of virulence factors that cause disease. Staph cells produce and release the necessary quorum sensing signals continuously. When only a few cells are present, the signals simply diffuse away. But if the cell concentration in a single location is high, the signals can accumulate and be sensed by the cells themselves. When the concentration is high enough, that is, when a quorum is reached, its presence activates cooperative behavior among the cells and also causes them to increase the production and release of the signal for quorum sensing, creating a positive feedback effect. That is, the more quorum signals that the cells detect, the more of the signal they release. In this way, a large staph cell population achieves synchronous behavior that controls their gene expression. In each cell, genes are turned on to produce the virulence factors that attack the cell's host. Thus, through the use of chemical signaling, staph bacteria can control their behavior, turning on their attack mode only when sufficient bacteria are present to overwhelm the host's defenses and successfully cause disease. Staph aureus, that is usually a harmless skin bacterium, can turn itself into a source of deadly disease. Considering now organisms that live in water, we see immediately that their pheromones must have quite different chemical properties. There's no advantage here in being volatile at all. The signals can be released and then carried by motion of the water itself to reach their recipients. Good examples of pheromones in aqueous environments are chemicals released by fishes as varied as minnows, catfish, zebrafish, and many others. These fishes have specialized cells in their epidermis that make and store the pheromone. When their skin is damaged, typically during attack by a predator, these special cells are ruptured and the pheromone is released into the water. Nearby fish of the same species detect the signal through their olfactory systems and immediately change their behavior to avoid the predator. These changes vary with the species involved, but may include freezing in place, darting quickly away, finding shelter, or in a school of fish, tightening of the school to take advantage of safety in numbers. The pheromones carrying the warning signals are sometimes chemically different from species to species, although in some cases, species other than the sending species do respond to them. In those cases, the warning signal serves as an interspecific warning as well as a pheromone. The compounds involved are reasonably stable in water and are middle-sized, non-volatile, and water-soluble. It's obvious from everyday experience that chemicals are equally important in mammals. When your dog sniffs at a fire hydrant or a tree trunk, he's receiving a host of social signals left in the urine of previous visitors. Dog urine contains information on gender, reproductive state, individual identity, social status, and territorial claims of an individual dog. Your dog is learning all these things about earlier visitors to the sniffing site. He can also determine how recently the mark was left, that is, which of several overlapping marks on the hydrant is most recent. There was early interest in studying mammals, but work here proved more difficult than in insects. Unlike in insects, mammalian behavior is influenced by a host of factors. In many insects that have attractant sex pheromones, if the male's olfactory system is destroyed, he shows no interest in the female. His sexual behavior is an obligatory response to sensing the appropriate attractant and other pheromones. In contrast, mammalian behavior is typically controlled by a multitude of factors. Human sexual response is obviously complex and influenced by many things. For example, unlike insects, anosmic humans, that is, people who have no sense of smell, can have perfectly normal sexual behavior. This more complex situation makes identifying mammalian pheromones and responses to them more difficult. For this reason, behaviorists were pleased to discover a suitable candidate for studying mammalian sex pheromones. This is the Syrian golden hamster, a 7-inch long solitary rodent native to northern Syria and southern Turkey, where it lives in burrows in the sandy soil. Behaviorists discovered that the male hamster is totally dependent on his sense of smell for sexual activity, so this becomes a useful species for studying sex pheromones released by female mammals. The female hamster goes into estrus, that is, becomes sexually receptive, every 4 days, and has a gestation period of only 16 days. On entering estrus, she lays several trails out from her burrow, marking each trail by touching her anal area to the ground repeatedly as she moves along. A male hamster that comes upon such a trail can follow it back to the female's burrow for sexual interaction. We became interested in learning what the female attractants was. It took about a year for a research fellow to identify the main attractant. He learned that the secretion deposited by the female along her trails was a mixture of about 60 volatile compounds. The entire mixture was required for full attractant activity, but a single component accounted for about 60% of the attractiveness. It's also the first in a suite of pheromones released by the female hamster to direct the male's reproductive behavior. On reaching the female in her burrow, the male approaches her head to head. He then moves to her side, where he sniffs and licks. He apparently finds another signal there, and this sends him directly to her anal area. He sniffs and licks here, and guided by a third signal, he mounts the female. We did identify this third signal, now called the mounting pheromone, as a small protein. So the golden hamster's reproductive behavior is controlled by at least 3 different pheromones. A noteworthy point concerns an important difference in properties of the 2 signals we did identify. The attractant involves small volatile molecules that travel through the air from the female's trail and can be smelled and followed by the male. In contrast, the mounting pheromone is a protein that is roughly 200 times the molecular weight or size of the attractant molecules. Molecules of this size are not volatile, so the male will receive this signal only on licking the female where it is secreted. This is an example of a chemical signal detected by taste rather than odor, and it makes good sense. A mounting signal that was volatile would drift away on the air and necessarily deliver its message to males whether or not a receptive female was nearby. This could lead to a chaotic situation, with males seeking but not necessarily finding any suitable females. This brings up the interesting question of whether there are sexual pheromones active in humans. Even if a response to them is not obligatory, might there be natural attractants that play a role in human sexual response? The perfume companies certainly would like to convince us that the answer is yes, and that they know just what to include in their attractive products. Unfortunately, a clear scientific answer to this fascinating question is not yet available. There have been numerous studies and suggestive results, but nothing more. We should note, however, that it has been known for some time that in general, volatile chemicals play important signaling roles in humans. A newborn baby, for example, requires only a day or two to recognize the odor of its mother's breast and to distinguish it from all others. Similarly, a new mother quickly learns to distinguish her baby's odor from that of other infants. In recent decades, there have been many investigations of mammalian pheromones, and we now know how important these signals are in directing the lives of many species. Many signals have been identified both behaviorally and chemically, and there are increasing investigations into the associated physiology, genetics, and neurology. That is, how the external signals are detected, how they trigger neural signals to go to what parts of the brain, and what messages are then sent where from the brain. We'll not go into details, but mammalian chemical ecology is rapidly becoming integrated into our overall biological understanding of mammalian species. To give you an idea of the importance of mammalian pheromones, let's look at the range of chemical signals that are currently known to be active in a mouse's life. Pardon? A mouse's life. Female mice undergo first estrus, that is, first become sexually attractive, at the age of about 4 weeks, and have a roughly 4-day estrus cycle. We know of several different pheromonal effects on this activity. Group-housed females respond to male odors by synchronizing their estrus cycles. This means they all become ready for mating at the same time. Second, male odors can accelerate the onset of puberty in juvenile females, promoting growth of the uterus and initiating estrus, again making them ready for mating as males are present. Third, female mice housed in a group of 8 to 10 per cage or exposed to the odors of group-housed females display estrus cycle delay, that is, postponing estrus until males are present. Overall, then, the odor of male mice influences female maturation and beginning of estrus to coincide with the presence of males. Also, pregnant female mice emit a signal that suppresses ovulation in subordinate females, and in addition indicates to males that they are no longer fertile. This presumably reduces sexual harassment and reproductive competition in the group. Male mice and receptive adult females are attracted to each other by volatiles in the urine of the opposite sex. In addition, there's a pheromone in male mouse tears that causes the estrus female to assume the receptive position known as lordosis. That is, standing still, arching the spine, raising the head and hindquarters, and lifting the tail, all to invite and facilitate copulation. A newly mated female will terminate her pregnancy, that is, undergo a miscarriage, on being presented with a strange male or his odor. In a natural setting, a stranger present in the female's accustomed territory would generally be a new dominant male that had defeated a resident male and taken over his territory. The female's behavior then allows her to mate with this new, strange, and now presumably dominant male that is now in charge. Termination of pregnancy does not occur if the second male is already known to the female. She can recognize individual males and responds favorably to a new dominant male. In a number of cases, investigators have shown that these various effects of a male on female behavior do not occur if the male has been castrated. Whatever the male signals are, their production depends on the presence of testosterone. Mice are born blind, and newborn mouse pups find their mother's nipples from chemical signals. Washing the nipples hinders suckling behavior, and destroying the newborn's olfactory cells in the nose leads to their death from failure to feed. Also, the odor of her newborn mouse pups is important to the female's recognition of them. Another set of mouse pheromones is involved in individual recognition and aggression toward foreign intruder males. Both male and female mice will attract intruders to defend their territory, their offspring, and their mates. Resident males will attack foreign males, but not foreign females or male castrates. Painting male castrates with intact foreign male urine or its purified constituents restores aggressive behavior in resident males, indicating the source of the signals leading to aggression. At least 2 of these urinary pheromones have been identified chemically, but they are active only in combination with additional recognition signals. Apparently, recognition in mice is complicated. These examples show that mice are quite dependent on pheromones for their everyday lives. The same is true of many other rodents, as well as more distant mammals such as foxes, elephants, or rabbits. For all of them, pheromones are woven into their very existence. Pheromones also occur in plants, and although perhaps we do not often consider that plants communicate with one another, this is actually quite common. In recent years, there have been increasing investigations of these plant signals. An instructive investigation in corn illustrates the use of both chemical signals and chemical defenses in plants. The investigation employed several corn plants growing near one another but not touching. The investigator touches the leaf of one plant with a soft brush, simulating the arrival on the leaf of an aphid. Aphids, which are common corn herbivores, are small, sap-sucking insects and are among the most destructive pests that attack cultivated plant crops. They number about 5,000 species in one large taxonomic family. Incidentally, aphids themselves make considerable use of the chemical information they receive from a specific plant in choosing to attack or avoid it for feeding. About 4 minutes after being touched with the brush, the corn plant responds in 2 ways. First, internally it sends chemical defense compounds to its touched leaf to repel the presumably attacking herbivore. At the same time, it releases volatile organic compounds into the air. These travel away from the plant and reach one of the nearby corn plants, where they alert this second plant to the danger. On receiving the volatile signal, the second plant responds defensively as the touched plant did. It too sends chemical defense compounds to its leaves, just as if they had been directly under attack. All it takes to activate a defense response in a group of corn plants near each other is touching one leaf of one of the group. Other examples of this sort of plant defensive signaling are known. Sagebrush attacked by herbivores release a volatile signal to warn other sagebrush plants. They respond by sending defensive chemicals to their leaves. Lima beans undergoing attack by spider mites send volatile signals that cause nearby lima bean plants to turn on the expression of defensive genes. Similar behavior occurs in plants as diverse as rice plants and poplar trees. Plants also use pheromones for purposes other than defense. 2 nice examples involve 2 different processes: synchronous flowering and synchronous seed production. When plants produce many seeds or flowers simultaneously, seed predators or herbivores can be overwhelmed, and many seeds or flowers survive simply because the predators cannot eat them all. In order to take advantage of this so-called predator satiation, some plants send signals to other members of their species to coordinate their flowering or seed production of the entire group. Many plants flowering at the same time also has the advantage of attracting more pollinators to a rich selection of flowers, and so increasing the likelihood of successful pollination. Now turning to interspecific signals, we see that chemical communication between species is also important all across the biological spectrum, from bacteria, fungi, and plants to invertebrates, fishes, and mammals. Here the signals can have 3 different effects, depending on whether the signaler, the receiver, or both benefit from the interspecific signal transmitted. The venom that kills or paralyzes a snake's prey clearly benefits only the sending snake. As an interspecific signal, a mouse's odor only benefits a predator, such as the hunting house cat that uses the signal to find and catch the mouse. Of course, this same signal as a pheromone does serve to the essential purpose of identification for the signaling mouse. Finally, a mutually beneficial signal. This is a flower's fragrance that attracts a foraging honeybee to stop and gather its nectar. The flower also benefits from the insect's visit by having its pollen transferred onto the bee's body and then carried away to pollinate a nearby flower. We can look at examples across various species to see the important roles these chemical signals play. There's a genus of parasitic fungi known as zombie fungi, owing to the effects their chemical signals have on their hosts. One species of this fungus affects carpenter ants with extraordinary results. This is an example of an interspecific signal that benefits only the sender. The zombie fungus begins life as a spore that is blown around with the wind. If it's lucky, it either lands on a carpenter ant or else could be picked up by the ant on the floor of the forest. Once inside the ant, the spore develops into a fungus that begins to secrete its chemical signal to change the ant's behavior. This ordinarily social insect begins to show a diminished ability to communicate and participate in foraging tasks with other ants. Next, it loses motor coordination and balance, and it fumbles its way around like it was drunk. Then the infected ant abandons its nest and climbs nearby vegetation. Once it reaches an elevated position, it latches onto the underside of the vegetation substrate with its mandibles. The fungus then kills the ant by sucking up the remainder of its nutrients inside and forms itself into a fungal fruiting body. This develops a new spore, which in its elevated position is able to catch the wind and so blows away to complete the life cycle of the zombie fungus. One of the longest known chemical signals that serves as an interspecific signal comes from the bark of a South American tree. When the Spanish colonized Peru in the 16th century, they found the local people regularly treated fevers with the bark of this tree. The Spaniards sent batches of the bark back to Spain, where it soon became the first really good treatment for malaria, and its use spread over the known world for 300 years, and it was in that time the most effective treatment for malaria. In the 19th century, the special chemical responsible for the bark's effectiveness was isolated and named quinine. In the bark's host, which is several Peruvian trees in the genus Cinchona, this bitter, toxic quinine protects the tree from bark-eating herbivores. A common noxious weed here in the Pacific Northwest is known as spotted knapweed, which was introduced from Eurasia with alfalfa in the late 19th century. It's very effective here in taking over large areas of grassland and rangeland at the expense of the native grasses growing there. In addition to its very large seed production and the long life of its seeds in the soil, and another important reason for the success of this plant is the production in its leaves and shoots of a volatile chemical called niacin that it secretes to inhibit germination of native grass seeds and so reduce competition for spotted knapweed. Interestingly, in its native habitats, spotted knapweed disseminates much less niacin than it does here, where it is invasive. At home, it has perhaps become accustomed to the surrounding competitive plants and does not attempt to prevent their growth. Or perhaps the competitors are immune to the knapweed's inhibiting signal, and so it no longer secretes it. Here, on the other hand, where the surrounding plants are not ones it evolved with, spotted knapweed makes a serious effort to restrain these other newer competitors. Similar differential behavior is known in other invasive plants and demonstrates an ability to tell nearby plant species apart. A nice example of interspecific signals that benefit the receiver to the regret of the sender are the chemicals that bring mosquitoes to attack humans. Gravid female mosquitoes must have a blood meal before they can lay their eggs. The primary attractant that draws them to their meal of humans is the carbon dioxide that we exhale. Mosquitoes can detect carbon dioxide at a distance of up to 30 feet from its source and then direct their flight toward its increasing concentration. As they move up the concentration closer to the source of the gas, mosquitoes are further attracted by several chemical compounds in human sweat and human skin oil until they finally arrive to feed on their human prey. For an instance of mutually beneficial interspecific signals, we can examine the interaction between a tree and its resident ants. Central American bullhorn acacia trees secrete a rich nectar that attracts several species of local ants. They come to feed on the nectar and remain to live in the many hollow thorns of the tree. Both the ant and the tree profit from this arrangement. The ants feed well and have a safe place to nest, and in return, they protect the tree by attacking herbivores to keep them away. And they even discourage other trees from growing too near the host tree by cutting off the intruder's leaves. A mutually beneficial situation exists in Africa between certain acacia species and their local ants. An interaction between dogs and humans provides interspecific chemical communication that may be familiar to dog owners. In a carefully executed experiment, humans watched either horror films or comedy movies. Afterwards, the axillary, that is, underarm, sweat from the two groups was collected to give pooled samples, respectively referred to as either fear sweat or happy sweat. The investigators then applied the sweat to cotton pads and allowed the dogs to sniff the sweat from one group or the other without the humans being present. The dogs used were Labrador retrievers and golden retrievers. Dogs that sniffed the fear sweat showed increased stress-related behaviors, including elevated heart rate, seeking the proximity of their owners, and various avoidance behaviors. Dogs that sniffed the happy sweat were more relaxed, interacted more readily with strangers, and displayed more exploratory behavior. Apparently, through their long evolutionary association with humans, dogs have developed a response to human emotional states with appropriate behavior of their own. There's some chemical information available concerning the axillary chemicals associated with particular human emotional states, but the specific chemicals acting on the dogs have not yet been identified. Mammalian prey-predator interactions provide numerous examples of several sorts of interspecific signaling. Predators like wolves, coyotes, and lions can follow scent trails of prey species like deer and antelope. Of course, these signals are not intentionally produced by the prey for communication, but are nonetheless effective interspecific signals. In the opposite direction, many prey rodents, such as mice and rats, detect predator odors from, say, cats, foxes, and weasels, and then modify their behavior by reducing their activity, increasing their vigilance, and avoiding dangerous areas. Once again, these odors are not intentional signals, but are compounds that vaporize from predator urine or feces or glandular secretions. Turning to crop plants, there's a set of hidden underground interspecific signals completely unknown to most people that are absolutely necessary for many crops to survive and thrive. These messages that pass between the roots of the plants and the fungi in the soil. Corn and tomato plants provide a typical example. When these plants become low on phosphorus, which is a chemical element indispensable for their growth, their roots release a chemical signal to attract certain soil fungi. The fungi move to the plant, releasing an answering signal that identifies them as being friendly species of fungus. On recognizing this message, the plant activates genes to allow the fungi to enter into its root cells under tight control. This controlled colonization creates a safe zone where the fungi deliver phosphorus and other nutrients to the plant. In return, the plant provides sugar to the fungus so it can maintain itself within the plant root environment. This mutually beneficial arrangement allows the corn and tomato plants to thrive. For some plants, selective breeding has produced varieties with increased sensitivity to these interspecific signals, leading to increased incorporation of fungi in the roots, increased nutrient uptake, and so increased crop production. Another set of consequential underground signals concerns nitrogen in the soil. Because plants must have nitrogen to survive and grow, millions of tons of agricultural fertilizer are applied to keep the soil rich in this element. An environmentally more favorable way to maintain soil nitrogen is to employ legumes, which are essentially peas, beans, and lentils. Unlike all sorts of other plants, legumes actually put nitrogen into the soil rather than take it out. By alternating cultivation of crops that remove soil nitrogen with that of legumes that replenish it, it is estimated that 30 to 100% of the requirement for nitrogen fertilizers can be avoided. Globally, legume-based systems put roughly 50 million tons of nitrogen into the soil annually. From our point of view, the interesting thing about legumes providing nitrogen to the soil is that it is really soil-dwelling bacteria that are responsible for the effect. An exchange of interspecific signals between legume roots and certain soil bacteria bring the bacteria into the roots where they form nodules. Inside the nodules, the bacteria live in protected environment and receive nutrients needed from the plant. The bacteria then convert atmospheric nitrogen into ammonia, some of which the plant employs in synthesizing amino acids and other chemicals required for its growth. At the same time, some of the ammonia passes from the legume roots into the soil, enriching it. This agriculturally valuable system depends entirely on the interspecific chemical signals that bring soil bacteria and legume together. Before ending our survey, we should look at how humans have developed commercial applications of some chemical signals. The Internet offers pheromone traps from several commercial sources for about a dozen household pests, including carpet beetles, clothes moths, rice weevils, and many other insect nuisances. A different sort of commercially available signal that should please pet owners is the dog-appeasing pheromone. Like many other mammals, a lactating female dog secretes this signal from glands located near her nipples, beginning 3 or 4 days after the birth of her puppies and ending a few days after their weaning. The puppies pick up the pheromone as they suckle, with the effect of relaxing and pacifying them and helping them to establish a bond with their mother. Now, for any pheromone to be commercialized, it's necessary either to isolate the signal in a large quantity from its biological source, or to substitute for this natural material a chemically synthesized replacement prepared in the laboratory. The dog-appeasing pheromone turned out to be a simple mixture of derivatives of organic acids that could be prepared by chemists. This synthetic version is sold to reduce anxiety in puppies related to sound sensitivity and fear of fireworks and thunder, and to control stress reactions in puppies enrolled in socialization classes. It's also been incorporated into dog collars and sprays that have found some success in treating anxiety-related problems. Similarly, there's also a commercially available pig-appeasing pheromone that is said to reduce aggressiveness in pigs and promote conflict avoidance. Interestingly, lactating female mothers produce a human-appeasing pheromone that is chemically similar to the dog version. I know of no effort to commercialize this signal, but a synthetic preparation has been studied for its possible utility in treating patients with severe psychiatric disorders. Results are promising, but very preliminary. Probably the most widespread human use of any kind of chemical signal is the application of insect pheromones as a means of environmentally friendly pest management. An important example of this in Washington state concerns the gypsy moth, an insect that is native to Europe and parts of Africa and is a seriously invasive species in the northeastern United States. Gypsy moth caterpillars are voracious feeders that strip leaves from whole forests and weaken or kill them. According to the U.S. Forest Service, the gypsy moth is one of the most destructive forest pests ever introduced into North America. These invaders are spreading across the United States from east to west and are not yet found in number in Washington state. The state, however, carefully monitors their possible introduction and presence here. Every summer, the State Department of Agriculture places small paper or plastic traps on tree trunks in forested areas throughout Washington, generally about one every square mile, but more in high-risk areas. Each trap contains a small amount of the gypsy moth attractant pheromone. Every few days, the traps are checked to determine if any gypsy moths have been attracted to them and trapped there. In this way, the state conducts a census that can locate any population of insects for destruction. Traps were installed every summer in the San Juan Islands when I lived there, and the fortunate finding was that no gypsy moths were ever counted in the islands. Another method of pheromone control of an invasive insect is also important in Washington state. This invader is the codling moth, which is well established as the key pest in the apple orchards of eastern Washington. The larvae cause significant damage by tunneling into fruit, feeding on the core and seeds, and of course rendering the fruit unfit to be marketed. Most varieties of apples are heavily damaged without some sort of protection from larval attack. While the use of insecticides in this regard became established in the late 1940s, the alternative employment of pheromonal control is environmentally more attractive and indeed now is in use on roughly 90% of orchard acres, of apple acres. This involves releasing and maintaining a high concentration of the female's attractant pheromone throughout the orchard during the whole mating season. This disrupts the mating process by confusing the male moths and preventing them from finding females for mating. If the males detect the attractant everywhere, there can be no telltale trails of vapor leading them to receptive females. Our last example is a different sort of human application of a chemical signal. A pheromone well known to beekeepers is the mandibular pheromone of the queen honeybee, a signal that regulates essentially all aspects of worker bee activity in the hive. It induces the presence of a permanent retinue of workers around the queen, suppresses the development of worker ovaries, inhibits the construction of queen cells, and delays the maturation of workers into foragers. In queenless hives, it can mimic the presence of a queen, and so the hive can continue to operate until a new queen is in place. It calms a colony that is preparing to swarm or has recently swarmed. A commercially available synthetic version of this mandibular pheromone is useful to beekeepers in keeping hives orderly and smoothly functioning. It is readily available right on the internet. In closing, chemical signals are nature's invisible language, exploited by all sorts of living organisms to survive, thrive, and interact with one another. From bacteria coordinating their behavior through quorum sensing, to alarm signals released by injured plants, to pheromones guiding the behavior of entire insect colonies, chemical signals represent one of biology's most ancient and universal means of communication. These signals help coordinate complex systems without words or wires. just revealing how interconnected and finely tuned nature is. The next time you catch the scent of a flower or notice how a dog responds to certain smells, remember that this is merely a small drop from an enormous ocean of chemical communication that has been shaping life on Earth for billions of years. Understanding these chemical languages not only deepens our appreciation for the complexity of nature, but also opens doors to new applications in medicine, agriculture, and conservation, reminding us that in the grand symphony of life, chemistry sometimes conducts the orchestra.
We have an opportunity to ask some very pressing questions here. Ed, you got a mic there? Yeah, right behind you.
What are these chemicals made of, and how large are the molecules?
Which chemicals are you talking about?
For the
Pheromones? Well, as I said, most pheromones are volatile molecules, which means they're fairly small, C6, 7, 8. I did give an example of a protein pheromone, which was detected by taste rather than smell. But in general, they are, as I said, volatile and relatively small.
I have a very practical question
Oh, good.
for Horizon House gardeners. The new gardens out here, which are all in little containers, are being attacked by bunnies.
Bunny rabbits?
Bunny rabbits. Now, in the beginning of your talk, you talked about tobacco being a repellent. If our gardeners got themselves a package of cigarettes and broke them up and scattered the tobacco in amongst their plants, would it help to repel the bunnies?
I haven't the faintest idea, but I hope you try it.
We will definitely let you know.
Certainly tobacco is odoriferous and easily smelled from a reasonable distance, and bunny rabbits have a well-known good sense of smell. So yeah, do try it. Let me know what happens.
Okay, we will.
Norm? Oh yeah. John, first John. Yeah.
Okay. Again, this is another very basic question because I'm not really a scientist in this regard.
It's okay, John.
We used to talk about all this mating behavior being instinct. Is there such a concept as instinct anymore? Or does this pheromones sort of takes that away?
Oh, I don't think pheromones take the idea. I didn't know there was an issue about instinct going away, but I thought instinct existed, and I don't think there's any problem about pheromones being active as well. I don't see a conflict.
A conflict, okay.
Norm?
I'm a Canadian as well as an American.
It's okay.
And I noticed when I crossed the valley in British Columbia that all the pines were dead. They were infested with this beetle that kills pine trees. But here in America, we don't have any of that.
Well, it's not true. There's lots of areas in California of
but they don't
dead trees. Yeah,
they don't wipe out a whole valley. I crossed valleys that were brown with dead trees.
I understand, and the extent of the area affected certainly will be variable all over the place. I don't think the Canadians have been especially mistreated by bark beetles.
Go to Colorado.
Two things. Two things. To John's point, in my mind, instinct is connected to the pheromones. It means the same thing. I mean, this is what instinct means, responding to these pheromones. That's how I see it. But anyway, as a person who is loved by mosquitoes, I was confused by the fact that mosquitoes are attracted by carbon dioxide, because I have always been with people who mosquitoes don't touch and who bite me. Is there an explanation for that?
I understand the issue, and I'm one of the ones that doesn't get bit. We all are obviously exhaling carbon dioxide, so my guess is that there's a effective difference in the other compounds. As I said, when the mosquitoes get closer, they're attracted by things in sweat and skin oil. And I've always assumed that I was lucky that my sweat and skin oil didn't have the right things, or not so much of the right things, but I don't really know more than that.
Many years ago, decades, almost century, when I was at UCLA, there were quite a few Arab students. At that time, UCLA was the attractive university because the climate was very similar to the Middle East. So at that time, the Arab students, males, would not use deodorant. I mean, it was just universal. You could smell them rows and rows and rows ahead of you or behind you. And so one time, a group of us asked them, we even brought a deodorant stick, saying, well, do you mind, this is something. And the answer was, no, they will not use it because their smell attracts women. So it didn't do it at UCLA. Maybe it did it in the Middle East. But that was their perception. That was their method of procreating.
Yeah, that's frustrating, isn't it? The repellent effect, of course, is well known, as you say. I suspect there are also attractants in both male and female sweat that work across both ways. I don't have a good answer, but I see their point.
I have a question. I was traveling in the wild area of the Missouri River with a person who is a chemist, and she said that some trees have developed an antibody to this bark beetle, and she felt that it would be smarter than cutting down whole valleys of trees to prevent their advance, to penetrate trees and see whether they have this capacity to resist this beetle. And she was she had a big argument in favor of that activity, but was very frustrated with the BLM and the forestry department for being unwilling to take the time and employ the people to do that, to just leave the trees that have the resistance to help. And so that made me think a lot about when I moved into my house in West Seattle, I had 3 flowering cherries, and they were separated. Their limbs didn't touch, but I'm sure their roots did. And one of them died from some disease that they get. And then the second one died. And then the third one went through a patch of losing its leaves, but then came back into full bloom. And I've always imagined that there was a chemical defense system that developed in that third tree from, is that kind of a crazy assumption, or is
No, I know nothing about your trees, but there's certainly, plants have been investigated, and that certainly is true in some cases. It is not universal, but it's a perfectly reasonable idea, sure.
25-year period of time?
Yeah, well, yeah, but in general, yes, there is communication, extensive communication among plants, particularly of the same species, and the fact that it can be defensive in that way is perfectly reasonable. Whether or not it's true in that case, of course, I have no idea.
Bill, thanks so much for a wonderful talk, but I have a question. Something has really intrigued me about these communication. I'm over here.
There you are.
Hi.
I knew who it was, but not where you were.
When you're sending messages out, there is an intended audience for which, and like for an example, a bird gets up in the spring and sits on the top of a tree and sings, "I got a wonderful territory." Every hawk within a mile knows exactly where it is. So I'm wondering with pheromones, the same problem exists. You send that signal out there and there's an intended audience, but a lot of other individuals that are negative that you don't want to hear what may hear. Do we have any evidence in the structure of pheromones that they're designed to make it hard for other than the intended audience to detect it?
By structure, you mean chemical structure?
Yes.
No, and I would have thought that would be unlikely. But you're the biologist. I can't imagine any structural feature that would have that very nice effect. I mean, obviously, if it could exist, it was the sort of thing that would evolve nicely. I mean, there's a lot of pressure for it. So, but I'm looking at it just from a structural point of view. I can't imagine anything to do with structure that would prevent predators from being able to do it. No, I don't see it. It's a nice idea. Yeah, but I don't think it's worked.
It's a nice idea from the point of the emitter.
Yes. Well, yeah, but that's who I thought we were in favor of. Yeah. Yeah.
Bill,
Yes, ma'am.
I'm wondering about interspecies attraction to these pheromones. If there's a little cross attraction there, and maybe, well, certainly repellent, but are there species that are attracted to other species' pheromones?
Oh, sure. Predators are attracted to. I gave an example of house cats are attracted to mice, and the mouse is sending out a signal that other mice use to identify the mouse.
Right.
But the cat picks it up as an interspecific signal to find the mouse. Yeah, I mean that's a simple example, but there are probably more interesting, complex ones. But that sure, that's real. Yeah. There's no earthly reason why that wouldn't develop evolutionarily. Yeah.
Okay.
Yeah.
So I have a question that's sort of related to the one that Gordon asked, and that is that we know that there are molecules that define self and non-self, and these are involved in organ transplant rejection, etc. Is there any evidence that these molecules lead to some kind of inter-individual signaling, for instance, to either promote or prohibit interbreeding?
Are we talking about interspecific or intraspecific? Intra?
Yes.
Yeah, sure. There's plenty of evidence that that mice can identify other individuals and do so regularly.
Yeah, but what are the signaling molecules?
I'm the faintest idea. I mean, yeah, that is a nice research problem.
I mean, those cell surface antigens.
Sure, but
floating away.
I think that from a chemical point of view, it sounds like a rather difficult research problem because you're talking about something that's produced at the individual level, which means you're going to have to work with one critter at a time to find it. And yeah, well, it's going to take several postdocs, several years. Is that? I mean, I know you would understand that. So, yeah.
Bill, I have a question. We know that among animals can communicate through these signals, threat to one another, correct?
Threat?
Threat, you know.
Well, yeah, prey animals get signals from predators.
Yeah. So is it, how about with humans? Can Humphrey Bogart really say, "I smell a rat"?
I always thought so.
Okay. Any other question? Bill?
We have a whole industry built on the idea that some odors are better for humans' attractiveness than others. Is there a kind of a golden goal that people are shooting for that particular pheromone that will universally attract the right people to you?
I once got involved briefly with a perfume company that desperately wanted to find a human attractant, human sex attractant. As far as I know, they didn't succeed. They paid me a reasonable amount of money, but it didn't do them any good.
One final.
But yeah, they're very interested. Yes, Norm?
I wonder why somebody hasn't worked to develop something anti-pine beetle. It's so destructive. North and south of the border, it wipes out valleys full of pine trees. Why hasn't somebody developed an anti-beetle?
Well, I don't know that the answer is they haven't tried. I mean, there are a lot of insecticides. I suspect it's an economic issue rather than a scientific one. I mean, there are plenty of insecticides available to wipe out pine beetles, and they are used. They are obviously not used as much as you have described these whole valleys of dead trees. But I don't know any other answer than yes, one has developed lots of insecticides, and it is certainly reasonable to work on developing one that is specific to pine beetles. But I doubt that would be very easy to do. But it's something one could think about doing. But I again suspect that the answer is largely economic.
Okay, I'm afraid we're going to have to wind up. We're running out of time. Thank you, Bill, very much. Thank you for your help.
Thank you.